Yay! It's the last topic in topic 2, photosynthesis. I have found that most students have a pretty good understanding of photosynthesis, but we're going to go through more of the details of it, light versus dark, and then just kind of like with enzymes, looking at limiting factors or factors that change the rate of photosynthesis. So going again back to what you know about enzymes is that there's optimal temperatures, pHs, and situations where they work the best. The same is true of photosynthesis. So we'll be looking through those different factors that affect photosynthesis rates and just a little bit more of like the nitty gritty of where it occurs, why it occurs, and the different pigments included. All right, so get ready. Photosynthesis. Yay. Okay, all right. Let's start out with just the basic equation. Again, something that you've probably seen before. Maybe you still remember it, maybe not, but let's make sure we have it in our notes, right? So photosynthesis is that the general overarching idea is that it takes in carbon dioxide, that plants take in carbon dioxide, that it's useful for plants, right? So we exhale, in cellular respiration, going back, like the CO2 is a product, right? And it is created as waste, goes out into the atmosphere. Plants are able to take that in and make it into a part of the living or organic matter, right? So it's able to take inorganic matter, right? Carbon dioxide and water, and in the presence of sunlight and chlorophyll, right here, I get the product of glucose, which is a sugar. Talked a lot about. We get a production of oxygen gas, and then water as a product as well. So water is actually on both sides of it. Right? You do need to know the amounts. If you did any stoichiometry with balancing, you probably, I don't know, used to doing this. So knowing just in general carbon dioxide, water, and the presence of light and chlorophyll makes glucose, oxygen, and water, but also knowing that C6O2 plus 12H2O, right, equals C6H12O6, which is our glucose molecule, with plus 6 molecules of O2 gas, and then the water. Right? It does require a photosynthetic pigment. We'll talk about some different types of pigments, and the plants have that organelle, some bacteria, right, are photosynthetic bacteria, but that are able to do this. We must have that organelle in order for photosynthesis to occur. Okay, just to compare the two, right? Hopefully you see or have seen that before, that they're like opposite reactions, that the products, right, the things that are created from one are the reactants of the other, right? And it kind of creates that circular, right? So if cellular respiration, which is produces energy, and it breaks down and has carbon dioxide and water, and then the photosynthesis takes those and it stores the energy, right? So the opposite. So I have cellular respiration releasing the ATP, photosynthesis storing that energy into that glucose molecule, right, with those bounds. So that energy that's stored is released by cellular respiration. Animals, right, cannot synthesize or make their own food in this glucose, so we depend on food, right? We have to consume them mostly in the form of plants, right? But we'll talk about some other types of food in our next unit. Okay, going to a little bit back about review about the light spectrum, maybe from physics, maybe from, I don't know, Roy G. Biv, right? Red, orange, yellow, green, blue, indigo, violet. The visible wavelengths of light go from 400 nanometers, yay, nanometers, woohoo, bet you missed nanometers, to 700 nanometers, so violet to red. This goes from the longest to the shortest. If you remember your EM spectrum, right above this, right above this, you've got UV light, so ultraviolet light is anything under 400, and then after 700, you get your infrared, right, which is heat, right, in the form of heat. Both of these not visible. This is our visible light spectrum, and you do need to know those wavelengths and the order in which they go in, right? The red and the orange. It's going to help you in a second, or it's going to help you memorizing these when we look at the factors that affect photosynthesis. Okay, all right. So chlorophyll. Going back to those organelles that we talked about in topic 1. We should practice drawing chlorophyll again, your favorite thing. But remember when we looked at like the mitochondria versus the chlorophyll, the thylakoid membranes were stacked in those circles, versus the mitochondria has the cristia that is folded on each other. So just know that there's different pigments that actually perform photosynthesis, but the main photosynthetic pigment is chlorophyll, right? And chlorophyll fills up our chloroplast, right? So that's how I remember the difference between those two. The chlorophyll fills up the chloroplast. The chloroplast is the organelle. The chlorophyll is that pigment that absorbs it. So remember, or try and access your prior knowledge and what you remember about light, is that we see what is reflected. So plants actually reflect the green light. So it's not good if we put plants under a green light if we're trying to increase photosynthesis, because that's the light that is reflected. So chlorophyll actually absorbs red and blue lights most effectively. So we're looking at photosynthetic rates. Those are the two where we're going to increase photosynthesis the most. If you think about the plant setup I have in class, it's under red light, right? So that's why I don't know if we've ever talked about that, but this is something that's going to increase my rates of photosynthesis. The other pigments that you are actually seeing in beautiful fashion right now are called carotenoids. Those are the different colors that we see when chlorophyll is broken down. Maybe we're going to go on a field trip to look at some different colors and talk a little bit about what happens during the seasons when chlorophyll is no longer needed. So there are other pigments that absorb wavelengths, but to maximize their photos, they absorb different wavelengths, and that whole process actually increases the process of photosynthesis. All right, so here's some pretty graphs to go through that, right? So you're looking at two different things here. So here's all my different types of pigments, the carotenoids, chlorophyll A and chlorophyll B. You're looking at the wavelength and the amount of light they're absorbed, right? So we can see right here, like 450 is like the peak for chlorophyll B. Chlorophyll A peaks here at a little bit before that and a little bit after, and my carotenoids are actually closer to 500, right? So over here, you're looking at so look, now the light color is the light color is here on the actual line. And this is looking at the wavelength and then the amount of light absorbed. So I see the most amount of light absorbed, just kind of putting those things into graphic form, right? At blue and red, which is what we talked about on our last one, right? I see the least amount right here where I have my greens and even closer to those yellows. So that is where I get the least amount of light absorbed, and thus I'm going to have the slower rates of photosynthesis. If I go back, so let me go back to the slide before, and this was showing how you can actually look at the different colors in plants. Who was it, Ellie? I don't remember. Someone was telling me that they did this with a paper towel, looking at how the pigments are absorbed. So this is called process called chromatography. It's a process that shows the different pigments that are in something. That solvent is the thing that we are dissolving. And in this case, we're looking at in a leaf, right? So the pigments, those leaves, you crush them up and then you dissolve it in a fluid. So it makes it into, something that can move. And then it's passed through a static material. Like I said, some people are talking about doing this with paper towels. This is a fancy little strip here. And because pigments are different weights and different sizes, they actually move at different speeds and will actually separate. So you can actually look at the different pigments in a leaf by looking at this chromatography. This sounds like something we should do. This sounds fun. And you can actually calculate what's called the retardation factor. Retardation means to slow down. And it's looking at the distance the pigment traveled divided by the distance the solvent traveled, right? So looking at the farthest that it travels, in the top, we're looking at how far this certain pigment. So you can actually calculate what's called the RF value for each one of these pigments based on how far they traveled up that static material, right? The material that they have to move through. Okay. All right. We do need to know the two different stages of photosynthesis, which are called light dependent and dark dependent. Light dependent and light independent. So things that require light, but then things that will happen even in the dark, right? So photosynthesis keeps on occurring in trees, but they're broken into two different processes, kind of like anaerobic and aerobic, right? You just kind of need to know the difference between them. So light dependent over here, this is kind of the ones that you have our general understanding of photosynthesis. The light and water go together and they make oxygen. This happens in the thylakoids. So those are those stacked membranes that you are used to drawing. This one right here, the light dependent, it converts that light energy into the ATP, right? This is the one that is converting that. The light independent, does not need light, right? This is the one this is where we actually synthesize those carbon compounds, right? So this is where this C6O 6H, blah, blah, blah. The glucose molecule is built. I'm tired right now. And so knowing that the difference between those two, the light dependent happens in thylakoids, the light independent happens in the stroma, right, to a different part of the plant, and the difference that are made. So we need light and water going into here. We need carbon dioxide going into this process. So these are all my reactants. And then here are all my products, right? The oxygen is made in light dependent. That actual glucose molecule is made in the independent. And these are our two forms of energy we've already talked about, the ATP, the adenosine triphosphate, and the NADPH. These are the different types of energy that are needed based on, that's going back to cellular respiration. All right, here's a fancier chart if you want to put this into your margin. Just takes basically this one right here, right, and puts it into a flow chart where light energy is needed, where the chemical, right? So for these two, this is the light independent. And then we're looking at what is created, right? What is where it goes, those molecules, and then the different types of energy that are needed. All right. I'm trying to look through here. Light is absorbed by water, split into, this is actually looking at when light is absorbed and what happens to the water molecule, right? That it's split into oxygen and hydrogen, and that oxygen is created as the waste, the O2 gas, right? My hydrogens are used to create my glucose over here. The ATP and the hydrogen NAHCP are used in the light independent reaction, right? So this is where that energy is actually required when it's, and think about that, that's where it's going into the glucose molecule and being stored. All right, here's the opposite, the light independent reaction, right? So that diffuses into chlorophyll. Just go ahead and pause this. I'm not going to repeat all this again, but go ahead and pause this and put this into your margins as well as a kind of flow chart. And make sure we're looking at, here is my product here, the organic compound, versus over here, my products are the oxygen and that water waste. All right, so make sure you pause it here, copy down this chart, and just as a summary of those last two slides. Okay. All right, limiting factors, temperature, light intensity, and carbon dioxide. So I'm going to go through all of those. And again, kind of think about with enzymes, those factors that affected enzymatic rates, right? And actually, you have a couple of the same ones. So think about, my temperature. My temperature is definitely one that changes the speed at which the reaction is going to occur. Light intensity, right? So how much light is available, and then how much carbon dioxide. That matters because that is it's a reactant. So I need carbon dioxide to make that glucose. So if I don't have enough carbon dioxide, it's going to slow it down, right? This is almost kind of like your substrate concentration, how much stuff you have to break down, right? So there's a very favorable condition where these three are like at their optimal level, and that's what we're going to look at in our next slides. Okay. All right. The first one is temperature. So photosynthesis, so why these things have these different rates or optimals, because they're controlled by enzymes that are used for carbon fixation, right? So one of them is called rubisco, and it's needed to fix the carbon, right, from the air. And so there's an optimal temperature at which that enzyme will have hit its peak, right, the rate of photosynthesis. Going back to something that we've already talked about, right? The kinetic energy of the enzyme, the number of collisions. So this optimal temperature is where I'm getting the most collisions of the carbon dioxide into that enzyme, and thus I'm going to increase the rate of photosynthesis. If it's too high, right, or too low, I'm not going to see that enzyme helping out. And so we don't get photosynthesis occurring in those two, right? Because either it's denatured or changed shape, so it cannot fix that carbon dioxide. That's a good review of what we've been going over in enzymes. All right. The other one is light intensity. This is, again, looking at the substrate concentration. So as I have more light, I'm going to reach a point right here where all of my chlorophyll, thylakoid membrane, they're working. And whenever they all are working, it's not going to go past that point, right? So after they are all working, just like in enzymes, when all of those enzymes are working, this is like the highest rate. So I'm looking at the rate of reaction. Okay, so going back through here, the light is absorbed. As the intensity increases, I have more chlorophyll working, but at a certain point, all of the chlorophyll are working. So I'm not going to get that happening any faster because everything that's working is possibly working is already working. All right. And then the last limiting factor I have is carbon dioxide concentration. Again, same thing as I said, like as I have the carbon dioxide coming in, but then I get everything working. So no matter how much more I have after that, it's going to level off, right? So more carbon dioxide means more compounds being made. It's going to plateau because all the enzymes responsible are already working, right? Those workers are already working at their max. Okay. All right. Here's the last slide. Looking at experiments. We're going to talk about these a little bit in class, but these are ones I want you guys to be thinking about for your papers, right? So possibly looking at photosynthesis experiments to talk about in your IB. Especially, there's been some really cool things. I'm kind of going off, but like algae being used outside of alternative, like places that create a lot of pollution or carbon dioxide. There's been some really cool experiments looking at how much carbon can be captured and where it can be captured. So how we can increase the amount of photosynthesis so that we're taking more carbon dioxide out of the air and decreasing that human activity. All right. And that, my friends, is the end of photosynthesis and the end of topic 2. I know I've talked really fast through that thing. So please make sure that you go back and listen. But I'm hoping that you have like your general knowledge of photosynthesis will help out a lot, especially with those products and the reactants. And then also what we've learned about enzymes just flows perfectly with everything we're looking at and the rate of reaction for photosynthesis. We'll look at these in light. I'll post the practice quiz. But that is it. That's it for new information. I will see you guys soon.